Hi class, we're going to pick up with section 17.3, which covers the production of formed elements. And so in other words, this is like how all of the blood cells get made. And I'm just going to switch to screen share mode really quick. Click all these buttons to do that. They should make it easier. Anyways, all right, so let's talk about how these formed elements are created. So obviously, one of the major sites of blood cell production is in the red bone marrow, which we recall probably from last semester. Hopefully you recall that from last semester. The whole process is called hemopoiesis. The prefix hemo or heme means blood, or is referring to blood, and then poiesis is referring to the making of. So this is literally translating to the making of blood. All the formed elements start from hemopoietic stem cells, yes, they're stem cells, or HSCs. So these HSCs, which are called hemocytoblasts, are the basis for creation of all of the blood in our body, all the blood cells in our body. Some general characteristics of all stem cells. They're all self-renewing, which means when they divide, through the process of mitosis, they're creating a new stem cell, and they're creating, a different type of cell from that. So when they divide, they're actually creating two different cells, a new stem cell and then a cell that's going to be differentiated into whatever the tissue is, in this case, a blood cell. So they're what we call a multipotent stem cell, which can give rise to different types of cells. So that new cell that's created that can differentiate. It can differentiate into any type of blood cell. It can be a white blood cell, it can be a platelet, it can be a red blood cell. So at that stage, it hasn't been determined yet what exactly it's going to be when it grows up. So, and they basically divide and like I said, they produce those two types of stem cells. They produce the stem cell that is going to recreate more stem cells, and then the stem cell that can differentiate into a new blood cell. So those hemocytoblasts produce the lymphoid stem cells. The lymphoid stem cells are responsible for production of all of the lymphocytes. So lymphocytes are named because they are tissues or cells that are going to basically migrate out to all of the lymphoid tissue in the body, and that's where they continue their development. So they meander through the body as stem cells until they get to the lymphoid tissue. Lymphoid tissue, and we have a whole chapter on this. This is things like the thymus, the spleen, the lymph nodes, right? The myeloid stem cells actually stay in the bone marrow, and they're going to differentiate into all of the other types of blood cells. So here you can see this process all in the red bone marrow. Where those nutrient arteries are is a very nutrient-rich area within the bone marrow. That's where this cell division occurs. The hemopoietic stem cells differentiate into lymphoid stem cells or myeloid stem cells in the process. So let's talk about lymphoid stem cells first. Lymphoid stem cells basically differentiate into something called a lymphoblast. The lymphoblast then becomes a pro-lymphocyte. It's this whole chain reaction of division after division after division, and then the pro-lymphocytes eventually become lymphocytes. That prefix pro usually means before, right? And this is all driven by colony-stimulating factors, or CSFs. Colony-stimulating factors are basically little local hormones that are within the lymphoid tissues themselves that are basically released. They can activate these lymphocytes, they can activate the immune response, they stimulate blood cell formation. So they have multiple roles, but their main job is to stimulate this differentiation in the lymphoid tissue. So here you see the whole process. We start with the lymphoid stem cells, go to the lymphoblast, then the pro-lymphocyte, and then the lymphocyte. So all of this kind of first two-thirds or first quarter of development is all happening in the lymphoid tissue, and then they differentiate in the, then they go into the blood once they're a lymphocyte, right? So the other type of stem cell that was created, again, we had myeloid and lymphoid. The myeloid stem cells are going to basically mature into something called a blast cell. So myeloid stem cells can differentiate into three types of progenitor cells. They can differentiate into monoblasts and myeloblasts. They could also differentiate into megakaryocytes. Megakaryocytes, if you think of the prefix mega, that they're basically like a humongous cell. They have lots of cytoplasm in them. Basically, the whole function of a megakaryocyte is to break off those pieces of cytoplasm, and those pieces are the platelets. So platelets aren't actually a fully formed cell. They're pieces of a megakaryocyte. So, or they could differentiate into pro-erythroblasts. So the lymphoid stem cells are always going to differentiate into lymphocytes. Myeloid stem cells are going to differentiate into monoblasts and myeloblasts, or megakaryocytes, or pro-erythroblasts. You follow me? What I think you should do before you flip to the next slide, or before you look in your book at the answer, see if you can make a graphic organizer of this, right? You start with your original stem cell, your hemocytoblasts, and see if you can differentiate it, lymphoid stem cells, myeloid stem cells, and then from there, right? That's going to help assimilate this information in your head. So here it is, this whole picture, right? You had the myeloid stem cells, and myeloid stem cells can be broken into the myeloblasts, megakaryocytes, or pro-erythroblasts. Those myeloblasts, again, all stimulated by these colony stimulating factors, can differentiate further, right? Myeloblasts and monoblasts, megakaryocytes, pro-erythroblasts. The pro-erythroblasts are actually stimulated by something different than a colony stimulating factor. They're stimulated by a hormone called erythropoietin, EPO. Erythropoietin is a hormone that is released from the kidneys, which we'll talk about a lot later. So these myeloid stem cells, basically, as you saw in that last slide, they can be monoblasts. Monoblasts differentiate into pro-monoblasts. Myeloblasts differentiate into 3 different types of myelocytes. These myelocytes are going to become a type of band cell. Sorry, I have an itchy nose. A band cell, right? The whole process here is to create the different types of blood cells, right? That's the whole purpose of this process. Pro-erythroblasts are going to go through several erythroblast stages and become a reticulocyte. So again, that graphic organizer that you have, see if you can follow these pathways just from the words. And then when we get to the pictures, which is on the next slide, of the actual words, break it down, double check your answers, fix things, or pat yourself on the back because hopefully you did it right. So here you go. The myeloblast and monoblast, the monoblast is going to become a pro-monocyte, and the myeloblasts are going to become band cells. Those band cells should look familiar. Remember lab, right? These are going to eventually be those 3 types of white blood cells. Megakaryocytes, boom, that's done. They break off and form platelets. Pro-erythroblasts are going to go through several different stages and eventually become a reticulocyte. Notice what is missing between the erythroblast stage and the reticulocyte. It loses something. Do you see what it is? So let's talk about the function of erythropoietin. Erythropoietin is generally abbreviated EPO. It is a hormone that's released into the blood plasma by the kidneys, and the kidneys are going to release this in response to basically low blood levels or low oxygen levels or hypoxia. So when your blood, and the kidneys have a very, very dense capillary network, but when the kidneys sense that there's low oxygen or low blood levels, they're going to secrete EPO. The EPO is going to stimulate your red bone marrow to differentiate and form more red blood cells. This whole process of erythropoiesis, right, making red blood cells. So, and that all happens in the bone marrow. So EPO is going to be stimulated under conditions of like anemia, decreased blood flow to the kidneys, maybe decreased oxygen, either from disease, high altitude, right, or any damaging that occurs to the lung surface, which is going to eventually affect your oxygen content in your blood, all going to cause the release of EPO from the kidneys. So let's talk about these myeloid stem cells, right? These myeloid stem cells form the pro-monoblasts, which become the monocytes, or the 3 band cells. The band cells are going to differentiate into the neutrophils, eosinophils, and basophils. The megakaryocytes basically shed their plasma like a little, I think of them like cotton candy almost, right? You got cotton candy and you pull off those little pieces. little pieces of cotton candy are the platelets, right? And then the reticulocytes, which become erythrocytes or red blood cells. So here's the continuation of the process. The pro-monocyte becomes a monocyte, and then our band cells differentiate into our 3 types of white blood cells, and the megakaryocytes make platelets. Reticulocyte eventually becomes an erythrocyte. And there's the whole image in all. So let's start from the very beginning. A very good place to start. Remember that? Sound of Music? Okay, sorry. This is coffee. I've been drinking it for hours, which is why my face is kind of red. So we have our red bone marrow. Our red bone marrow is going to be stimulated by various hormones and factors to undergo erythropoiesis or undergo hemopoiesis, making blood, right? Those hematopoietic stem cells can differentiate into lymphoid stem cells and myeloid stem cells. This, by the way, if you've been following my directions, should follow along with the graphic organizer that you've already made. Hopefully. So the lymphoid stem cells can differentiate into lymphoblasts, right? This is all happening in the lymphatic tissue, right? In our lymph nodes and our thymus and our spleen. This is happening outside of our bone marrow, right? So lymphoid stem cells migrate out of the bone marrow to these lymphoid tissues. That's where this process of lymphopoiesis occurs, where we create a pro-lymphocyte and then a lymphocyte. And even lymphocytes differentiate even more. We'll talk about that in a few chapters. So myeloid stem cells form progenitor cells. These 3 types of progenitor cells, we have monoblasts and myeloblasts. This is all stimulated by CSF, colony stimulating factors. The monoblast will differentiate into a pro-monocyte and eventually monocyte. The myeloblasts are going to form band cells, which form our neutrophils, eosinophils, and basophils. And then our megakaryocytes will eventually form platelets, right, like cotton candy. And then our proerythroblasts from our progenitor cells go through several different stages, a reticulocyte, and then eventually a fully formed mature red blood cell called an erythrocyte. That's all stimulated by EPO, which is released from the kidneys. Summary, right? So let's talk about platelets. They're cell fragments, so they're not specialized. They're formed from megakaryocytes. They function in the blood clotting process. They're continually replaced through just natural processes and this natural process of renewal that occurs in our red bone marrow. So very small. Abundance range. Do you need to know numbers of these things? For lab, yes. For a lecture, not so much. Most of the lecture questions that I'll ask you about these cells will they will cue you in. So I will say something like, so-and-so has, this number of white blood cells or this number of neutrophils, which is high or which is low, right? So I'll kind of give you a cue as to, exactly what's off with the numbers. You don't have to know specific numbers. So you can pause the video here. You know what? I'm actually going to stop the video here for this little section, and then I'll start a new one for the next section, okay? And as always, let me know if you have any questions. Bye.